Hello, this is Lecture 4D, the final lecture for Week 4 of Applied Immunology. This will complete our module on T-cell biology for this course. We will end this topic today with a discussion of T-cell activation and differentiation through lymph node priming, as well as the various effector functions of CD8 and CD4 T-cell subsets and how these contribute to adaptive immunity, and end with a brief overview of the formation of long-term T-cell memory. If we revisit our generic immune response diagram that we established at the beginning of this course, we've filled in a lot of details this week about how this diagram specifically applies to T lymphocytes. We know about the structure of the TCR and the specific signal transduction pathways that are activated downstream of TCR binding to peptide MHC on antigen-presenting cells. Today we will round out these details by learning more about the effectors that are produced by activated T cells, how these vary amongst different subsets of T cells, and how these promote pathogen clearance. As a general rule, direct cytotoxic mechanisms are typically exerted by CD8 positive T cells, while indirect mechanisms that act on other cell types are mediated through helper CD4 positive T cells. Let's first recap the events that lead to immune priming in secondary lymphoid organs. From our lectures on innate immunity, we've learned how infection in local tissues, such as an epithelial surface, can lead to the activation of resident antigen-presenting cells. This example from the textbook looks at Langerhans cells, which are just a specialized type of skin-resident macrophage. So we've learned that upon exposure to infection, APCs such as macrophages and dendritic cells are exposed to PAMPs that activate pattern recognition receptors, such as toll-like receptors, and that PRR signaling activates the APC. This activation leads to the phagocytosis of microbial antigens that are then processed and presented on MHC class 1 or 2, and the upregulation of other activation molecules, such as co-stimulatory ligands like CD80 or CD86. Importantly, APC activation also triggers expression of a chemokine receptor, including something called CCR7. Remember that chemokines serve as chemotactic cues that can direct the migration of immune cells throughout the body. CCR7 binds to chemokines named CCL19 and CCL21, which are expressed in the T cell zone of lymph nodes. So once the activated APC drains out of the skin and enters lymphatic vessels, CCR7 expression is an important step that allows the APC to migrate towards the chemokine gradient of either CCL19 or CCL21, which is established within lymph nodes. The APC then arrives at the closest skin-draining lymph node through the afferent vessel at the top of the lymph node. And again, the lymph nodes are organized into a cortical region at the top, and this is where afferent lymph arrives, and this overlies the B cell zones that are shown in yellow. Because CCL19 and CCL21 are expressed most highly in the T cell zone of the lymph node, the CCR7 expressing APC migrates deeper into the T cell region of the lymph node, and this is shown in the diagram on the right in blue. Once in the T cell zone, the APC can crawl around and sample different T cells, and this is a process called browsing, until it finds one that expresses a cognate TCR that recognizes its own peptide MHC complex. Naive T cells are those located in the lymph node that have not encountered cognate antigen before. We've established that once an activated APC browses and successfully locates a naive T cell with TCR specificity to its peptide MHC complex, there are three critical signals that are relayed between APC and T cell at their interaction surface, which again is called the immunological synapse. Signal 1 is TCR activation through recognition of foreign peptide MHC, and this leads to the signal transduction pathways that we covered extensively in Lecture 4C. In our last lecture, we also learned how signal 2, or co-stimulation between T cell CD28 and corresponding co-stimulatory molecules, is required for optimal TCR signaling and the survival of activated T cells. In naive T cells, signal 1 and 2 lead to the proliferation of that T cell clone, which is a process that we also refer to as clonal expansion. Note that the production of IL-2 and IL-2 receptor subunits downstream of TCR signaling is required for successful clonal expansion. Lastly, signal 3 is the T cell's detection of cytokines that are produced by the APC. Although cytokine signaling is not required for the survival of activated T cells, it does influence the differentiation of certain types of T cells, especially CD4 positive helper T cells. As we will learn in upcoming slides, the types of cytokines produced by APCs are determined by the type of pathogen that activated that APC. So cytokines are extremely important in fine-tuning the T cell response to most appropriately address broad categories of pathogens, and this is in addition to the specificity of the response that's encoded through the TCR. So signal 3 represents a key additional step in the initiation of pathogen-appropriate T cell responses. This entire process of successful activation and differentiation of naive T cells in secondary lymphoid organs is called T cell priming or immune priming, which we've introduced a few times before in previous lectures. There are a few other ways for T cells to become activated, but for the purposes of this introductory course, we will focus on naive T cell priming in lymphoid tissue as the primary mechanism of T cell activation. Once T cells are primed, they then need to leave the lymphoid organ and traffic to infected tissues where they can exert their effector functions. This process involves a chemotactic lipid called sphingosine 1-phosphate, or S1P, that's shown here in the diagram in green. S1P is present in high concentrations in the efferent lymphatic vessels, which again are the vessels that exit the lymph node and allow cells to re-enter circulation. S1P levels are low in the lymph node tissue itself, so therefore an S1P gradient is established within this lymph node. When T cells here in blue are activated in the lymph node tissue, they transiently downregulate expression of the receptor for S1P, called S1PR1. Several days after activation, T cells have proliferated into many daughter cells that have acquired effector functions, so we refer to these as effector T cells. And effector T cells regain expression of S1PR1, which recognizes S1P, and this draws the activated T cells into the efferent lymphatics. S1PR1 expression dynamics therefore regulate the ability of activated T cells to exit the lymph node, and this allows them to re-enter vascular circulation and then zero in on the site of infection based on chemokine gradients that are established in locally infected tissues. In this diagram, note that FTY-720 is a drug treatment that can be administered which blocks S1P binding and therefore prevents activated T cell egress from lymph nodes. Once primed T cells successfully migrate or traffic to the site of infection, they can exert many different types of effector functions based on their cellular identity, which we often refer to as their T cell subset. We will go through each of these in more detail in the upcoming slides, but remember in general that CD8 positive T cells recognize antigen presented on MHC class I on all cell types, and primarily function through cytotoxic mechanisms that kill a target cell, though they can also produce inflammatory cytokines. On the other hand, CD4 positive helper T cells recognize antigen presented on MHC class II by professional APCs, and depending on the subset of CD4 T cell can produce many different types of chemokines and cytokines. CD4 T cells can be split up into Th1, Th2, Th17, and Treg subsets, as well as T follicular helper cells, which we will discuss shortly. And all of these differ with respect to the initiating signals that influence their differentiation, as well as the cytokines and chemokines that they produce. Although there's a lot of information presented here, for this introduction to T cell functions, I mostly want you to understand that CD4s primarily act through non-cytotoxic mechanisms. So they typically function by producing cytokines and chemokines that help recruit and activate other effector cell types in order to better eliminate certain types of pathogens. Before we get into the specific effector functions of each of these T cell subsets, I want to discuss some key differences between the activation of T cells during T cell priming versus their activation as effector T cells. We've already learned that naive T cells, shown here in purple, require TCR engagement plus co-stimulation in order to become activated during lymph node priming, and this can be viewed as the recognition phase of the T cell response. Upon the receipt of signals 1 and 2, the naive T cells then enter a proliferative state, which requires the production of the survival cytokine IL-2, as well as certain subunits of the IL-2 receptor that bind to this cytokine with high affinity. This is the process of clonal expansion, where T cells proliferate rapidly and also undergo differentiation towards different subsets, and this is based on whether or not they've received various cytokine cues from the APCs that are present in the lymph node. These proliferation and differentiation phases of the T cell response take several days in the lymph node, and at the end they yield high numbers of effector T cells that express the S1P receptor 1 and are able to leave the lymph node to traffic to peripheral tissues. Once effector T cells arrive at a target tissue, they are then able to exert their effector functions. But a key difference between naive T cells and effector T cells is the fact that effector T cells can become activated in response to TCR stimulation alone, so they no longer require co-stimulation or signal 2 in order to deploy their effector mechanisms. In this example, we are looking at CD8 positive T cells, which recognize foreign antigen on MHC class I that's expressed by infected cells. TCR activation alone is sufficient for the CD8 T cell to kill the infected target cell through cytolytic mechanisms that we will discuss shortly. And I want you to just think about how this makes sense. So the T cell was initially primed using a professional APC in the lymph node, which express co-stimulatory markers. But once that T cell differentiated into an effector T cell, it needs to be able to recognize any type of infected target cell and be able to eliminate it. This includes non-professional APCs, such as most other somatic cells that only express MHC class I and don't express any sort of co-stimulatory markers like CD80 or CD86. But keep in mind that these other types of infected cells still need to be able to be targeted by cytotoxic T cells in order to clear infection. This example uses CD8 T cells, but keep in mind that this feature of effector T cell activation is also observed in CD4 positive T cells. Even though CD4s usually don't kill targets, they may still need to help activate infected macrophages or B cells that are expressing antigen on MHC class II, but just haven't yet upregulated co-stimulatory marker expression. Let's start to learn more about the actual effector functions of T cells, beginning with CD8s, since in a way they're the most straightforward. Again, CD8s recognize peptide presented on MHC class I, which is expressed by almost all somatic cell types. MHC class I also presents peptides sampled from the host cytosol, so it typically presents antigens derived from intracellular pathogens such as viruses. Since virally infected cells are compromised and the best course of action would be to remove the survival niche for that virus, it makes sense that the primary function of CD8 T cells is to kill an infected target cell. CD8 positive T cells are also often called cytotoxic T lymphocytes, which is abbreviated as CTL. Now, effector CD8s kill infected targets by releasing cytotoxic molecules from intracellular organelles called granules, and these are kind of spat out at the target cells into the immunological synapse upon TCR activation. Following TCR stimulation in effector CD8s, granules accumulate at the immune synapse and then undergo calcium-dependent release of their contents. These contents include perforin, which forms pores in the target cell plasma membrane, and this allows other molecules to translocate into the cytosol of the target cell. Granules also contain granzymes, of which there are several family members, but all of these function by cleaving protein substrates in the target cell, and their activities lead to a form of programmed cell death called apoptosis. Granulysins are a form of antimicrobial peptide that are found in granules of human CD8s but not other species, and these can function by killing bacteria and fungi. CTL can also produce inflammatory cytokines such as interferon gamma, TNF alpha, and lymphotoxin alpha, and these can all further contribute to host defense, although the primary function of CTLs is to kill intracellularly infected host cells. Also keep in mind that CTL can perform this cytotoxic effector function on many different infected target cells, as long as they express cognate antigen on MHC class I. So we can think of effector CD8s as selective serial killers, in that they can target many infected cells in quick succession, but this killing occurs in an antigen-dependent fashion. And also, because granules are released at the immune synapse, they're concentrated towards the target cell that the CTL interacts with through its TCR to peptide MHC, and this restricts killing to just that single cell. And this is important because it spares uninfected neighboring cells in order to prevent excessive tissue damage or immunopathology. Let's next look at CD4 positive helper T cells, which can get a little bit more complicated because they can be differentiated towards several different effector cell subsets based on the cytokine signal 3 of T cell activation. If you think back to our lecture on innate lymphoid cells, you will remember that ILCs can be separated into different subsets based on the inducing cytokines produced by innate immune cells that trigger their differentiation. They also differ with respect to the effector cytokines that they produce and the types of infections that those effector cytokines correspond to. CD4 T cells can be similarly separated according to these parameters, although they're obviously different from ILCs in that CD4s require TCR signaling upon recognition of peptide on MHC class II, and so they therefore function in an antigen-specific manner. We can also add in more information about the signal transduction pathways that occur downstream of signal 3 detection in CD4s, as well as the types of master transcription factors that are required for CD4 differentiation. And these are called master transcription factors because they are absolutely required for CD4 differentiation towards that subset. Note that CD4 T cell subsets are abbreviated by the acronym TH, which stands for T helper, and it's followed by a descriptor for that specific cell subset. This adds up to a lot of information across 5 main subsets of CD4s, so let's go through each of them individually. CD4s can differentiate towards a T helper 1 or Th1 subset in response to vesicular pathogens, such as bacteria that replicate in intracellular vesicles. These are induced as a part of a type 1 immune response, which are typically deployed against intracellular pathogens. Professional APCs produce the cytokines interferon gamma and IL-12 in response to this, which is detected by naive CD4s during priming and induces signal transduction via STAT1 and STAT4, and these trigger the expression of the master transcription factor TBET. TBET then controls differentiation towards the Th1 subset, and this is characterized through the production of interferon gamma. So Th1s therefore amplify the differentiation of more Th1s through additional production of interferon gamma. And we will learn more about how interferon gamma also helps infected professional APCs defend themselves against vesicular pathogens. We'll cover that on the next slide. The next CD4 subset are Th2s, which are induced as a component of type 2 immune responses against parasites, including helminths. Naive CD4s that detect IL-4 from APCs trigger signal transduction through STAT6, which leads to the expression of the master transcription factor GATA3, and this regulates the expression of Th2 effector genes, such as IL-5 and IL-13, which are important for defense against parasitic infections. GATA3 also triggers additional expression of IL-4, which, like interferon gamma and Th1 cells, forms a positive feedback loop to help differentiate more of its own CD4 subset. Th17s are differentiated in response to infection with extracellular microbes, such as bacteria or fungi. These infections stimulate IL-6 and IL-23 production by antigen-presenting cells, and these are detected on CD4s during priming, which leads to the initiation of signaling through STAT3, and this induces expression of the transcription factor ROR gamma T. This is a nuclear hormone receptor that controls expression of the cytokines IL-17 and IL-22, although notably, unlike Th1s and Th2s, Th17s don't amplify their own differentiation through the production of additional inducing cytokines. That is, Th17s don't produce more IL-6 and IL-23 to generate more Th17 cells. IL-6 can also trigger differentiation of T follicular helper cells, or TFH cells. TFH cells can be induced downstream of any type of infection, so they are not pathogen-specific. TFH cells also require STAT3 signaling downstream of IL-6 detection, but they diverge from Th17 cells in that they are formed following expression of the transcription factor BCL6. BCL6 actually upregulates a chemokine receptor called CXCR5 on TFH cells, and this functions by keeping the TFH cells localized to the B cell follicles and secondary lymphoid organs. And this is because B cell follicles produce the CXCR5 ligand CXCL13. This is important because the function of TFH cells is to provide T cell help via IL-21 secretion, and this helps B cells optimize their antibody responses, which we're going to learn more about next week. So it's worth noting that TFH cells are different from the other CD4 lineages presented here, since they're not associated with a certain type of pathogenic infection, and they're also retained in secondary lymphoid organs rather than trafficking to sites of infection. Lastly, T regulatory cells, or Tregs, are an extremely important type of CD4 subset that is necessary for dampening immune responses rather than amplifying inflammation. Now, natural Tregs are a bit unique in this chart in that they are differentiated in the thymus during T cell development rather than in the secondary lymphoid organs like the rest of these subsets. Since Tregs are generated during thymic selection, they're not specifically induced in response to any one type of pathogen and instead have specificity for self-antigens. Tregs are derived from potentially autoreactive T cells. So if you remember back to this affinity model graph on the right from our lecture on T cell development, Tregs are represented by this green bar on the plot on the right. These are CD4 positive T cells that passed positive selection but have stronger affinity to self-peptide on MHC class II than the repertoire of T cells that actually recognize foreign antigen. And so this tendency towards self-recognition is paired with sensing of the cytokine IL-2 during development, and this leads to STAT5 signaling and the induction of the master transcription factor FOXP3. FOXP3 positive Tregs are an essential suppressive CD4 subset, that negatively regulates inflammatory responses by other leukocytes. And it does this through the production of lots of different types of immunosuppressive molecules. And these include the regulatory cytokines TGF-beta and IL-10. Tregs therefore function to block the activation of other immune cells, including the inflammatory activities of the other CD4 subsets listed here. So these cells are extremely important for promoting the resolution of inflammation following pathogen clearance, as well as in preventing autoimmunity. I know that this is a lot of information on CD4 subsets, but I want to emphasize the fact that any single naive CD4 clone can differentiate into most of these different effector subsets based on the cytokine milieu that's produced by professional APCs during T-cell priming. The exception to this is Tregs, which develop in the thymus. In this plot on the right, you can appreciate how a single naive CD4 could give rise to either Th1, Th2, or Th17 lineages, and that this is entirely dependent on which types of signal 3 are detected by the CD4 in combination with TCR and co-stimulatory signaling. Note that T cells responding to different types of infections express different chemokine receptors, here shown as CCR5 on Th1 cells, CCR4 on Th2 cells, and CCR6 on Th17 cells. These correspond to the types of chemokines that would be produced in response to local types of infections, and explains how these cells can find infected peripheral tissues following their priming in places like the lymph node. When we consider things like chemokine receptor expression and effector function, it really helps drive home the fact that T helper cell responses are specifically tailored in a way that best supports defense against the specific classes of pathogens that were responsible for initiating their differentiation in the first place. Now that we've established factors that play into the differentiation of various CD4 subsets, let's next learn about how their effector functions specifically address these types of initiating infections. So, unlike CD8 T cells, which act directly by killing target cells, all CD4 effector functions act indirectly by enhancing the activities of target cells that express cognate antigen on MHC class II. And again, this is similar to the function of ILC subsets, although CD4s differ in that they must recognize targets through MHC class II. So Th1 CD4s produce interferon gamma, in addition to some other things, and interferon gamma promotes macrophage activation and antimicrobial programs that can help them eliminate vesicular pathogens, including things like intracellular bacteria. Th2 cells produce IL-4, which helps to recruit mast cells and basophils. They also produce IL-5, which recruits eosinophils, and they also generate IL-13, which helps stimulate mucus production. All of these effectors are required for successful clearance of helminths at mucosal surfaces, such as the intestinal epithelium. Th17 cells produce IL-17 family cytokines that activate non-immune cells. These are called stromal cells, and induces them to produce growth factors and chemokines that facilitate the development and recruitment of neutrophils. Meanwhile, IL-22 stimulates antimicrobial peptide production by epithelial cells at barrier surfaces. And together, these effectors help eliminate extracellular pathogens such as bacteria and fungi. TFH cells are induced by all types of infections and localized to B cell zones in secondary lymphoid organs, and this is done in order to help optimize antibody responses. An important cytokine for this function is IL-21, which induces the proliferation and differentiation of B cells into a lineage called plasma cells, which are responsible for producing large quantities of antibodies. Since antibodies are an extremely important component of adaptive immunity against any type of pathogen, it makes sense why these TFH cells would be induced downstream of any type of infection. Lastly, regulatory T cells suppress inflammatory responses by both innate and adaptive immune cells, and this is done in an antigen-dependent manner. These effects include a lack of activation by target cells, such as other T cells. And again, Tregs are really important for promoting the resolution of inflammation, as well as preventing autoimmunity. One last property of T cells is their ability to form long-term immunological memory that protects the host from future reinfection with that same pathogen. So if you remember back to our introductory lectures, this is a key final goal of a successful immune response. Lymphocytes of the adaptive immune system are responsible for mediating this type of immune memory, and this is accomplished in part through the specificity of lymphocyte antigen receptors. Immune memory also requires differentiation mechanisms that yield stable pools of memory lymphocytes, and these are maintained long after the successful clearance of a given pathogen. So let's learn more details about how T cells accomplish this final goal of an immune response. Today, we've learned about how T cells are primed in secondary lymphoid organs by professional APCs, and that this leads to their proliferation and differentiation towards effector subsets that possess various functions. We know that these effector T cells may remain in lymphoid organs, as is the case with TFH cells, or they might need to travel to non-lymphoid tissues in order to exert their effector functions, as is the case most of the time with CD8s, Th1, Th2, and Th17 subsets. But what happens after the effector phase of T cell activation when immunological memory must be established? Now, the majority of activated effector T cells are short-lived effector T cells, or SLECs, and these will die by apoptosis following the successful clearance of infection. However, there's also a small subset of effector T cells that maintain expression of a key cytokine receptor called the IL-7 receptor, and this gives them a survival signal in response to the cytokine IL-7. So these IL-7 receptor positive effector cells are called memory precursor effector cells, or MPECs, and these are responsible for forming memory T cell pools after the resolution of infection. And just note that this occurs for both CD4 and CD8 positive T cell subsets. MPECs in humans express a surface marker called CD45RO in addition to the IL-7 receptor and can form a few different subsets of memory T cells. The first are central memory T cells, which express the chemokine receptor CCR7. Remember, this CCR7 is the same chemokine receptor expressed by activated APCs that allows them to home to the T cell zone of secondary lymphoid organs. So CCR7 expression on memory T cells therefore recruits these cells to lymphoid tissues. So central memory T cells are specifically found both in the circulation and in secondary lymphoid tissues, where they are retained after restimulation with a secondary infection. And the second type of memory T cells are those that don't express CCR7. So they are free to roam around the body in both the circulation as well as non-lymphoid tissues. And these are called effector memory T cells, which express both chemokine receptors such as CCR3 and CCR5, as well as beta-1 and beta-2 integrins. And so together, these integrins and chemokine receptors allow effector memory T cells to rapidly enter non-lymphoid tissues from circulation upon their restimulation with something like an infection. The third and final type of memory T cells are tissue-resident memory T cells, which are abbreviated as TRM. Similarly to effector memory cells, resident memory cells lack CCR7 expression, but unlike effector memory cells, TRM do not re-enter circulation once they've seeded a peripheral non-lymphoid tissue. This is due to the expression of CD69, which reduces S1PR1 expression, as well as the integrin CD103, which binds to a ligand called E-cadherin that's expressed in epithelial tissues. So collectively, CD69 and CD103 both function by retaining resident memory T cells specifically in that tissue site. So TRM are unique amongst T cell memory subsets in that they actually do not re-enter circulation. Each subset of memory T cells can coordinate rapid responses to reinfection with that same pathogen. So central memory T cells can proliferate and undergo clonal expansion in lymph nodes, while effector memory cells can immediately traffic to new sites of inflammation and infection in case they need to respond to a pathogen that previously infected that individual. Tissue-resident memory cells are optimally positioned to rapidly respond to reinfection with that same pathogen at the same tissue site where primary infection occurred. So taken together, all of these forms of T cell memory represent an extremely important component of long-term immunological memory that is mounted against specific pathogens. Now, I want to end this last lecture by taking a step back and placing T cell responses in context with some of the other mechanisms of innate immunity that we've learned about so far. We've learned about barrier surfaces and the immediate phagocyte responses to infection once those surfaces are compromised. And we've also learned about innate immune activation in response to local infection through things like pattern recognition receptors, complement activation, and antigen presentation. We've linked these innate responses to the initiation of adaptive immunity this week by learning about T cell priming at tissue-draining lymph nodes, and also by learning about how effector functions of T cells are then deployed at the site of inflammation in order to clear infection and then form long-term T cell memory against that specific pathogen. Next week, we will build upon this knowledge by adding B cells and their antibody responses to this picture as well. So I know that we've had a lot of dense lectures this week concerning T cell biology, but I just want to make sure that you try to center your focus on understanding how each of these components synergize together in the generation of a successful immune response. Upon completion of week 4 of this course, you should be able to integrate what we now know about T cell immunity along with other types of innate immune responses, and also be able to conceptually place each of these into the 5 main goals of the immune response as they relate to pathogen detection, immune cell activation, the deployment of effector responses, the resolution of inflammation, and the formation of immune memory. To summarize, today we've learned that the activation and differentiation of naive T cells during T cell priming leads to the production of T cells whose effector functions are most appropriately tailored to fight the type of pathogen that initiated the immune response in the first place. We've also learned about the different types of memory T cells and how these contribute to the long-term immunological memory that is a hallmark of the adaptive immune system. We've established the effector functions of cytotoxic CD8 T cells, which undergo activation into effector CTLs that leave secondary lymphoid organs to directly kill infected cells that are found in non-lymphoid tissues. We've also established that helper CD4 positive T cells can be differentiated into various subsets, and that CD4s act indirectly by producing cytokines which enhance inflammatory responses by other immune cells. The important exception to this are regulatory T cells, which are critical for negative regulation of inflammation. And together, these 2 primary categories of T cells are extremely important players in the adaptive immune system. This concludes the lectures for the Week 4 T cell module for this course. You have a reading assignment to accompany these lectures that's outlined in the syllabus as well as being posted to Canvas. You also have an extra credit weekly quiz and a discussion board prompt. So if you want to submit a response for this week's discussion board, you will be providing a brief summary of a disorder belonging to a family of pathologies called severe combined immunodeficiencies, or SCID. So please see Canvas for additional details. Good luck with all of your assignments, and I will see you next week for our module on B-cell biology and humoral immunity.